Okay, good morning, everybody, and welcome to your second class in human biology. This is going to be a little bit abbreviated because on Wednesday I went over time, so I'm just going to keep this one relatively short, and then we will get partway through this PowerPoint set, and then we'll finish the PowerPoint set on Monday. Okay, so today we're going to be discussing basic chemical concepts. Basic chemical concepts. Do not panic. If you find it difficult to memorize this the first time that you hear it, or conceive of it the first time you hear it, then redo the lecture, watch it again, and read a text. Your text, if you want to, probably has relevant information, but you can find the relevant information anyway. But remember that we're only doing the very basics. Don't wander off into the weeds. And again, I repeat to you, as I always do, those who read succeed. And reading is essential. Reading and writing is essential in the learning process. Please keep doing it. So let's talk first of all about atoms, about matter, and about how everything that we see around us is constructed. Because we're going to move on, after we've done some basic chemistry, we're going to move on to talk about biomolecules, the molecules that actually make up the human body. And in order to understand how those molecules are made, we need to understand some basic things about how matter itself is made. So the first thing to think about is an element. There are numerous elements on our planet, in our universe. Each element has unique characteristics. And if we look at an element, we'll find that the very smallest part of the element, which preserves all the properties of the element, is called the atom. So that is the smallest division that we can make of an element, which will have the same properties as a big lump of it. And atoms are composed of a nucleus, which is positively charged overall, and then circulating around that nucleus in quite a complex fashion, there are numerous electrons, and the electrons are small, and they have a negative charge. Remember that they carry one negative charge. And under normal circumstances, the number of positive charges in the nucleus equals the number of negative charges that are circulating around it. And the electrons that we are particularly interested in are the ones that are on the outside. And you'll see that they circulate in a kind of a shell around the outside. It's here described as a circle. It's often very much more complex than that. It's not necessarily a circle, but in any case, we can conceive it as being this shell around the atom. And all of the characteristics of the atom, and hence of the element, are conferred on it by the number of electrons that it has, and by the behavior of the electrons, especially in the outer shell. So remember that. All of the characteristics, all of its chemical behavior is going to be determined by these electrons, by the electrons in total, but especially by these in the outside. And you'll see why right now. We're looking here at an element called sodium. So here it is here. Here's its nucleus, and you can count up how many electrons it has. It has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Just because of the way the atoms are constructed, we're not going to discuss the rules. There are rules about how many you can have circulating around the nucleus in any one shell. We end up with, in sodium, with one electron that's scooting around here. Okay, so let's take another element. This is chlorine. Chlorine is a gas. And we have a look at its configuration. Let's see, here's its nucleus. Let's count its electrons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. Now there's something about atoms. That is, for all the atoms that we will worry about, they really like to be able to show to the outside world a complete outer shell of 8 electrons. 8 electrons. They strive to show that. It is determined by quantum mechanics and quantum physics, but that is, in other words, have a look here. Look at this, 1, 2, 3, 4, 5, 6, 7, 8. It would be quite advantageous for the sodium to be able to just show that shell. And in fact, this electron here is quite loose. It's far away from the nucleus, and it's relatively loosely associated with the atom. Have a look here at chlorine. We'll count out the outside shell. 1, 2, 3, 4, 5, 6, 7. It only needs 1 to present a complete shell of 8 to the outside world. So the way in which these elements behave is very strange. They are both of these, both sodium and chlorine, sodium a metal, chlorine a gas, are extremely poisonous and extremely reactive. And the reason that is so is because chlorine very, very vigorously grabs electrons from other things. And it grabs those electrons to put 1 electron in its outer shell. Sodium, on the other hand, is very, very, very reactive because it tries to ditch this. It tries to ditch its electron, its 1 lone electron, so that it shows 8 in the outside shell. Okay? Now think about this. I told you, and you need to remember, that an electron has a negative charge. So if an electron has a negative charge and I take an electron away from sodium, I end up with an atom which has a relative positive charge. It's lost a negative charge, so it has become slightly positive. And here it is here. And that we refer to when an atom has changed its electron configuration, gained or lost, we call it an ion. There it is there. There's the word there. So this is a sodium ion, and it has 1 positive charge because it lost an electron. What happened to that electron? Well, we can donate that. This sodium ion can donate that electron. It can donate it, for example, to chlorine. And that is exactly what happens in solution. In solution, it will happily lose this electron and donate it to chlorine. Now we have 1, 2, 3, 4, 5, 6, 7, 8 electrons in the outer shell. And because chlorine now has gained 1 electron, it is an ion, but it is a negatively charged ion. And here's its charge there. In solution, these 2 atoms can be apart from one another quite happily. But if I take the water away and I force these atoms close to one another, this doesn't grab its electron back again. This doesn't throw off the foreign electron. Instead, the positive ion is attracted to the negative ion and they stick together. And when it pull all the water away, they will crystallize out as a solid. And that solid is ordinary table salt. It's ordinary table salt. That is what table salt is. It's NaCl. That's its proper chemical name. And you'll notice something very, very important. Sodium was a metal, very poisonous, highly reactive. Chlorine is a gas, also poisonous, very, very reactive. Is salt poisonous? No. Is salt reactive? No, it is not. There's nothing for it to react because everybody's happy. The sodium is happy. The chlorine is happy. It's got 8. Sodium is happy. It's got 8 in this outer layer. And they stick together because one is positively charged and the other is negatively charged. They are bonded. They become bonded together. As long as there's no water, they are bonded together. If I put them into water, they dissolve very, very easily. That is a characteristic of a compound with an ionic bond. Dissolves very, very easily for reasons we won't go into. But there are specific reasons why that happens. This kind of bond where an element donates an electron and another one accepts, and they then stick together because of their charge, that is a specific kind of chemical bond. It is called an ionic bond. And it is very important in biology because this is how we acquire how we take in many solutions. This is how we take in sodium. And for example, we need sodium. We need this sodium ion especially, and in our functioning. And we take ionic substances in the form of solutions in water. So they are very important biologically in that way. This bond, however, is not that important in constructing biomolecules. Instead, to construct biomolecules, we need to look at this kind of bond. This is the second kind of bond to consider. Again, there are demands upon an atom imposed by quantum physics for what they aspire to in an outer shell. And now this I told you, it's 8 for sodium and chlorine. This is a much simpler atom. It has only one shell. And in the first shell of an atom, the desired number is 2 electrons. So that is, now we're looking at the simplest of all atoms. And it's, by the way, the very first atom that was ever created in the Big Bang. This was it. And this is hydrogen, a gas which you've all heard of, abundant in our atmosphere. And it has, here's its nucleus, and its nucleus just has one positive charge. And it has one electron circulating, but the quantum drive is to acquire 2. And hydrogen is indeed quite reactive. And if you put it together with another hydrogen atom and you provide some energy, you can bond the 2 atoms together. And now, although neither of them actually has 2 all the time, they share their electrons. They circulate in a cloud around both nuclei, and they bond the 2 hydrogen atoms together. And this is called a covalent bond because the ions spend an equal amount of time around each of the things. And we have not set up an ionic situation. Okay, each of them gains, each of them gives up to a certain extent. The sharing is equal, and that is why it is covalent. And this bond, in fact, is extremely strong. It is extremely strong, and this covalent bond becomes very important in biomolecules. Almost all our biomolecules, their primary structure is developed by forming covalent bonds, and that are strong, that contain, and those, remember something else that's important I told you. If I want to bond this hydrogen to this hydrogen, I can't just put them together and expect it to happen. I actually have to put in energy. So I have to put in energy to get this to happen. This is actually a store of energy because I put it in. And if I split these apart, it will result in a release of energy. So these covalent bonds are important because they're strong. They can build structure, but they can also contain energy. And that we're going to see that is really significant when we talk about things like sugars and fats and things like that, where the covalent bonds are a store of energy, which we can access. If we split those compounds apart, we can extract the energy and we can use it. Okay, so we've got 2 kinds of bonds, ionic bond, covalent bond. Make sure you understand the difference between the 2. we're going to spend a lot of more time talking about covalent bonds than ionic bonds. So the last kind of bond is not that simple to understand, but it's very, very, very important in biology and in structuring biological molecules, especially proteins, and then nucleic acids, which are things like DNA and RNA. So we have to understand a little bit about the hydrogen, this kind of bond, which is called a hydrogen bond. So I need to explain this to you. Have a look and you'll see. We've already met hydrogen, which has one electron. And many, many things use hydrogen as a source of an electron. And this element here is oxygen. And oxygen requires two electrons to complete its outer shell. Oxygen is very reactive. It's a very, very, on its own as oxygen, it is extremely reactive because it is always trying to grab these electrons from other elements. And this is the commonest and for us the most important compound which oxygen forms. And here it is here. Here's the oxygen, and it's gained one electron from a hydrogen there, and it's gained one electron from a hydrogen there. And it has formed this compound, two hydrogens and an oxygen, H2O. And you all recognize what that is. It is water. H2 is two hydrogens and an oxygen. Here's another molecule. When we put these atoms together, they form a molecule. They form a compound, but we refer to the individual unit as a molecule. So here's a molecule of water. Here is a molecule of water. Now, something happens when different atoms get together, especially something like oxygen. Oxygen is very, very, very attractive for electrons. It will pull electrons towards it very, very strongly. We say it is electron negative. It pulls electrons. So this bond here is actually a covalent bond. It's a very, very powerful, strong bond. It is very difficult to split water up. You have to put in a lot of energy to do it. So it's very difficult to break these bonds. It's quite difficult. Here's the oxygen. So it's got a hydrogen there, but it so powerfully attracts the electrons that the electrons spend more time around the oxygen than they do around the hydrogens. Remember that the electrons here are just circulating all the way around all of its components like this in a cloud. But they zip around the hydrogen and they spend a longer time around the oxygen. They zip around the hydrogen, spend a long time around the oxygen. And because they spend more time around the oxygen starts to acquire a bit of a negative charge. Here it's described as delta. And it's a delta negative. So think of that delta, it's telling you on balance, the oxygen is slightly negative and the hydrogens are slightly positive. This confers a very important property on something like water, because if I bring another molecule of water near to another one, the negative part of the molecule here, the oxygen, is actually attracted to a hydrogen. We'd also have one coming here. We'd have one coming here, one coming here, one coming. In case of water, they start to bond. They start to approach one another, and they form a very weak bond. It's not a strong bond at all. It's easily broken. But it is a bond, again, a bond of attraction between charges. It's not an ionic bond. It's just that this area of the molecule is slightly negatively charged. This area here is slightly positively charged. So they tend to be attracted to one another. And this hydrogen bonding is incredibly important in water because it gives water all of its properties. That is why water behaves as the way it does. It's why water forms ice, for example, when you freeze it. It's why water is such a very, very, very good solvent. We won't discuss exactly why, but all of the properties of water are because of this, its ability to form these hydrogen bonds, because of this differential of charge around it, etc. So this hydrogen bonding is important in water, but we're also going to find that it is critically important in the structure of biomolecules, of the large molecules that make up living tissue. And we're going to encounter reference to hydrogen bonding when we talk about proteins, as I told you, because it's hydrogen bonds that often give proteins their shape in space. And we're going to talk about it especially when we talk about DNA, because DNA is a double molecule, and the two halves of the molecule are held together by hydrogen bonds. One of the features of these hydrogen bonds is that they're very weak, but they're often very, 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 very numerous. There's a gazillion of them. And so when there are a lot of them, they start to impart strength just because they are so numerous, even though an individual one is easily separated. Okay, so remember those: ionic, covalent, hydrogen. Right, so let's have a look now very basically at the science of organic chemistry. And organic chemistry is actually the science of compounds that are constructed by carbon. And all living organisms, on our planet at least, all living organisms consist mostly of carbon-based compounds. There is a carbon skeleton to all of our biomolecules. And there's a really good reason why that is the case. Carbon is the one atom, the one element, which possesses a huge range of abilities to form different structures, with different shapes in space and with different properties. And we'll see exactly why in a second. Everything that we're going to talk about, proteins, DNA, carbohydrates, molecules, all of these things, they're all composed of carbon. They're all based in carbon. And I'll tell you right now that there's a very important reason for that. And that is that there is a source of carbon very easily available, which is carbon dioxide in the atmosphere. And it is the responsibility of the plants to take carbon dioxide out of the atmosphere, to take water and put them together into the basic biological compounds, which all living things, including us, depend on. So that is ultimately the source of the carbon. The source of the carbon is carbon in the atmosphere, which is transformed by plants. It is fixed by plants into compounds, which everything else can then depend on. We are heterotrophs. We depend entirely on compounds which have already been made, and we change them, we transform them, we eat them, and we transform them into different things. So when we look at biological molecules, we'll find the backbone is carbon, but there may be many other things associated. Most commonly, the most common elements associated with carbon in biological molecules are hydrogen, and then also very often oxygen, and then some other things as well, as we'll hear in a minute. But most organic compounds, at the very minimum, contain carbon and hydrogen. So here, this is carbon here. This is how its outer shell is displayed in space. It has 4 outer electrons. So that 4, what is the desirable? Is 8. That means it can form very, very easily, form covalent bonds. It does not form ions. It never gives up in normal reactions, does not give up its electrons or gain electrons, but it can very easily be induced to form covalent bonds in order for the carbon to gain another 4 electrons. And that means that each of these projected into space in this tetrahedron, each of these can bond with something to gain an electron, or in actual fact, we can take 2 of these and bond to gain or share rather another 2. That we'd call a double bond. Carbon becomes very, 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 very versatile. It's extremely versatile because it has these 4 electrons available for covalent bonding with other things. Now, the first thing that it can bond with is itself, is carbon itself. And if you look down at a pencil, if you've got one near you, look at that. And what you're looking at is carbon atoms all bonded together into a crystalline form. That crystalline form of carbon we call graphite. So that is one example. That is not an organic compound, but it's an example that carbon can bond to itself very, very easily. And when we look at carbon, especially in biological systems, we find that there are only a few, in biological systems, there are only a few other atoms that we usually have to consider. And the most important is hydrogen. That is the commonest. The next is oxygen. We'll see how carbon bonds to oxygen to form an inorganic compound first, but then I'll explain how that later on. We'll see that oxygen can also be bonded with carbon. It can bond with nitrogen. That is really common in biological systems. And as I've told you, it can bond with itself. And this is the foundation. The bonding of carbon to itself is the foundation of all biomolecules. So the first thing is, if we just take carbon, we can bond carbon together into long chains. So you can just have carbon, 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 carbon. And then we can add to that, we can add hydrogens. Remember, it's got 4 bonds, okay? So now picture this. We're going to take 2 carbons and we're going to join them together with a single bond, okay? And so we got carbon, carbon. How many electrons does it have available? Does each of those have available now? Or one of the electrons is being shared in the covalent bond. They have 3 sticking out. And onto those 3, we can bond hydrogen. And that, for example, is C2H6. That would form a compound. And because it only got carbon and hydrogen, C2H6, because it's only got carbon and hydrogen, we call it a hydrocarbon. And those are common biological compounds because that is fossil fuel. That is what our fossil fuels are. They are huge molecules. It's not just 2 carbon, carbon, 6 hydrogens. It could be 1,000 carbons and all of their attendant hydrogens. And those are organic compounds. All of our fossil fuels, they arose in living things. So there is an example where living things are creating these very long molecules just composed of carbon and hydrogen. Here's an example, the simplest example of carbon bonding to oxygen. And carbon bonds to oxygen with double bonds, carbon, double bond. So here's the carbon, it's got 1, 2, 3, 4. It's sharing to oxygen, 1, 2, there, 1, 2 there. This is carbon dioxide, CO2, carbon dioxide. That is important because it is our source of carbon. in the biological world, that is our source of carbon, is carbon dioxide. Here's an example of carbon bonding to nitrogen. Carbon usually bonds to nitrogen in combination with hydrogen or another compound or another element. And here is one here. There's a carbon, 1, 2, 3, 4. And it is bonding to oxygen, the double bond there, C double bond O. And here it is bonding to nitrogen with single bonds. The nitrogens then have bonds to hydrogen. So, and this is a compound, this is a simple organic compound called urea. And in human biology, very important because it's our primary waste product from proteins, from metabolism of proteins. And it's excreted by the kidneys in the urine. So this is urea, and for us, a waste product. This is just examples for you of some different ways in which carbon can bond to different things, just to demonstrate to you the versatility of the carbon atom. In terms of biological molecules, for us, the most important thing about carbon, though, is that it can bond to itself. Now, you don't have to freak out. I don't want you to, you don't need to remember all of these, okay? All that I want you to do is see some very basic things and then realize how versatile the carbon atom actually is in forming different shapes. So first of all, here is the compound that I described to you. 2 carbons, CC, with hydrogens. Each carbon, 1, 2, 3, 4 available electrons bonding to 4 other atoms. 3 hydrogens, a carbon, 3 hydrogens, and the other carbon. Covalent bonds, very, very strong. And when I make this compound, C2H6, its name is ethane. When I make this, I have to put in a lot of energy. I have to put a lot of energy in to create these covalent bonds. That is really useful because this is a stable compound. I can store it. I can store it for a long time. I can store it for eons. And I can then mix it. I can mix it with oxygen and I can burn it. And what I'll end up with is carbon dioxide and water, but I'll also end up with heat and light. The energy contained in these bonds can be released. It can be released as heat, it can be released as light, but under very special circumstances, it can also be released as useful biological energy. We can do this inside a cell in a very controlled fashion, and we can use that energy. We can use the energy contained in that bond. Now I want to ask you a question. I say we put in a lot of energy to make these bonds. Where does that energy come from? Just think about it for a minute. Where does the energy come from? Is it just floating around? Does it come from, it could come from other compounds, that is true, but it doesn't. In actual fact, the ultimate source of this energy is sunlight, because the first steps, taking carbon dioxide and water together and putting them into a compound where we have covalent bonds, that requires sunlight. It requires the energy of sunlight to make those initial carbon bonds. What we do with those compounds that are made afterwards, we may end up with things like ethane. But remember that, first of all, the source of the carbon is carbon dioxide. Predominantly, the source of hydrogen is water. The source of the energy here in the bonds, ultimately, the ultimate source of the energy was sunlight. Isn't that? And that's a marvelous thing. That is an amazing thing. Because when you think, okay, look at the next compound. This is this is an example of how we can start to add carbons together, one after the other. We can start to make long chains, very long chains. They can go very, very long. The next one, got ethane. This is propane. Right, so now remember what I said. These bonds here, the energy is actually originally sunlight. So we all use propane. So next time you fire up a propane grill, okay, put your hand there and have a good look. This compound propane was made millions of years ago. It was made by plant action millions of years ago. And the ultimate source of the bonds, the energy in the bonds, was the sun. Burn propane and look at the flame. You'll see light and you'll feel heat. And the light and the heat you feel is the light and the heat from that million-year-old sunlight. And it's been stored for you in the propane. It has been stored for all that time until you burned it. And it was reduced again to carbon dioxide and water. Its original components and its energy is released for us to use, but spare a thought for the fact that it has a very interesting origin. All you need to remember on this diagram, first of all, we can start to add carbons one after the other into long chains. In this case, all what we're doing is we're adding hydrogens. These are all hydrocarbons, okay, that we're looking at on this diagram. We can change the length of the molecule by adding carbons. If you look here, they all have single bonds, but you know, I can take that bond there, this hydrogen, I could actually take that and double bond it to this carbon here. That would also work. We'd have 2 bonds there, 1, 2, 3, 4. That is a double bond. It does something very important. These molecules here are straight, but as soon as you put in a double bond, it kinks the skeleton. And so when you look here, have a look here, here are double bonds. Where you see this, it's just drawn straight for us, but in actual fact, in 3-dimensional space, this molecule would have a bend in it. This one here, this is exactly the same, by the way. This has the same chemical formula. This one here, 1-butene and 2-butene have exactly the same chemical formula, but they have double bonds in 2 different places. They have 2 different shapes. And because they have 2 different shapes, they behave differently. And this is also a way in which we can start to use these double bonds to make shapes in space. So that they can carbon joined into molecules. They can vary by length. They can vary by double bond position. They can also have branches. So here, for instance, this is butane. Here's just straight, and that's the butane that we burn in cigarette lighters and things like that. This here is 2-methyl propane. You don't need to remember the names. It's just to show you it gets a different name. It has a completely different structure, but if you count up the carbons, 1, 2, 3, 4, and you count up all the hydrogens, you'll find they're just the same number of carbons, same number of hydrogens in both, completely different compounds, completely different kind of behavior, because the shape is different. The shape is different. Very importantly, in biology, we can also take these long chains of carbons and we can flip them around to actually form rings. And this is something we're going to see is tremendously important. For example, in things like the nucleic acids, we're going to see ring formations. It's also very important in the sugars and things. Very important biological molecules have rings in them. The 4 things that you need to remember: carbon skeletons can vary by length, double bond position, by their branching, and by the formation of rings. Okay, so we're going to stop there, and I'm going to start at this slide on Monday and try and complete as much as I can of this section on biomolecules on Monday. Okay, thank you.